Yeah, well, this isn't Andy, this is Juliet, so, well, carry on.
Okay. So Andy and I did actually liaise about our title. It's the only thing we liaised on, but it didn't actually make it into the program. We're going to be talking about embracing the complexities of scale and diversity in disease ecology. And I'm going to focus on the ecology of vector-transmitted pathogens from around 1993 to the present. As you'll see, I had to narrow even more than that in order to come up with anything tractable to say. But disease ecologists tend to like vector-transmitted pathogens because there are lots of fascinating interactions between the pathogens, the vectors, and the hosts. And these are a variety of different disease-transmitting organisms, from various species of ticks to triatoma bugs, which transmit Chagas disease, the vector of, this is Culicoides sonorensis, which I believe transmits blue tongue virus. Otherwise, it's closely related to the vectors of blue tongue. Mosquitoes, of course, and fleas. And I left out tsetse flies. But there are a wide variety of different arthropods that transmit infections. And it turns out that many of them have been studied in the past 20 years in the context of disease ecology. But I have, being in the session on disease ecology that is in the midst of all of these wonderful talks on human pathogens and livestock pathogens, I decided that I should be focusing on things that are involve wildlife, involve natural populations in their transmission cycles. So these are the two probably best studied vector-borne diseases within the context of disease ecology. And these are sort of publication trajectories or epidemics of publications, as Julia pointed out yesterday, for Borrelia burgdorferi, which causes Lyme disease, and West Nile virus, which obviously causes West Nile encephalitis. And as you can see, both of these diseases have had a lot of research done on them over the course of the past 20 or 30 years, but very few of the publications have actually been using dynamic models. So the red is the research papers through time associated with the left y-axis, and the blue is the dynamic modeling papers through time associated with the right axis, which of course has a much different scale. So even the best studied vector-borne diseases of wildlife are relatively under-modeled or underserved in the modeling literature. From this point on, I'm going to focus exclusively on mosquito-transmitted, or at least dipteran-transmitted infections, but primarily mosquito-transmitted infections in order to be able to focus, with apologies to people like Pete Hudson, who have done excellent work on tick-borne diseases of wildlife. I myself am relatively new to the vector-borne disease world and have primarily been concentrated on mosquito-transmitted infections. So following the meeting in 1993, Chris Dye and Brian Williams wrote a paper that was part of this famous blue book called Nonlinearities in the Dynamics of Indirectly Transmitted Infections, or Does Having a Vector Make a Difference? This paper essentially explored the pseudo-equilibrium approximation, which is basically a way of modeling vector-borne, vector-transmitted infections as if they were directly transmitted infections. And they used the Ross-McDonald framework, which we saw yesterday, and assuming a constant vector population focused on dipteran vectors, which are short-lived and mobile, they concluded that dipteran-transmitted infections probably meet the pseudo-equilibrium criteria, which is essentially saying that rapid convergence, that there's rapid convergence to the vector isocline, which justifies setting dy dt, or the change in infected but not yet infectious mosquitoes, and dz dt, or change in infectious mosquitoes through time, equal to 0. Which implies that the dynamics of diphtheria-transmitted infections are approximately equivalent to dynamics of directly transmitted infections. Those are, I would argue, some fairly strong assumptions. For one thing, vector populations are almost never constant. And of course, as the vector population increases, the prevalence is going to decrease, and vice versa. As the vector population decreases, the prevalence may go up or down depending on whether or not there's differential mortality in infected mosquitoes, which of course they addressed to some extent. So they had a list of potential nonlinearities that could produce exceptions to the notion that vector-transmitted infections could be modeled essentially as directly transmitted infections. And their list included things such as if the host population is regulated by a vector-borne infection, if they're saturated in the prevalence of vector infection, if vector mortality increases with the infection rate or the parasite load, and preferential biting on infected hosts. I think there were a few others. But so obviously, it's not always valid to say that that vectors don't make a difference, but sometimes you can say that that's a good approximation. Okay, so that's 1995, the output of the last Newton meeting. So now I want to focus for a few minutes on a systematic review of mathematical models of mosquito pathogen transmission from 1970 to 2010, published recently by a number of people, including several people in this room. The authorship list is so long that I don't actually even know all of my co-authors, so hopefully I'll meet you over the course of the week. But this was quite a monumental task. This was a review of mechanistic models of mosquito-borne pathogens. So it's a slightly broader categorization of models than the West Nile and Lyme disease curves that I showed you before, which was only including dynamic models. This is anything that's mechanistic, so it could just be a vector capacity or vectorial capacity or R naught equation that has been parameterized. This is, again, the epidemic curve of publications on mechanistic models of mosquito-borne pathogens. I said I was going to focus on infections transmitted with wildlife as one of the components of the transmission system. And you can see that these publications are really, truly dominated by malaria and dengue, which is no surprise because, of course, those are the ones that we're most interested in from a public health point of view. They're following the introduction of West Nile into the US, as you saw dramatically on that earlier curve. West Nile virus in general got a lot more attention in the research, in the literature, and of course in the modeling literature as well. And then the other category here includes Rift Valley fever, yellow fever, which does have some wildlife component, including in some of these models, chikungunya, Ross River fever, Murray Valley encephalitis virus, and Western equine encephalitis virus. And there are a total of 26 publications in the purple that are West Nile publications, and a total of 55 for those 6 pathogens in the other category. So relatively little attention. One of the things that we did, and really primarily Bobby and Alex, the lead authors on this paper, did, was after we had read and scored all of those 388 models, they classified them in terms of how different they are from the basic Ross-McDonald assumptions.
What is it, the number of models published in a specific year? Is that what
the previous, yes.
So the same model could appear in several
Oh no, it was models. Sorry, it's individual models. So if a model was reused, then it was not counted as a separate model. Yeah, but there weren't very many of those that didn't have at least some modification. Bobby and Alex developed what they refer to as the Ross-McDonald Index, which is basically just a count out of 15 possible ways that you could make reasonable assumptions that deviate from the Ross-McDonald model, sort of the most basic 15 assumptions that you might want to vary. Looking at these models, most of them are almost all of them are either identical to the Ross-McDonald model in their basic assumptions, although they are tailored to specific diseases and specific contexts, or they make one or two deviations. And of course, this is for all of those models, including malaria and dengue. And then breaking it up a little bit by what is different in the different diseases. So the color scheme here is the same. Purple is West Nile, and gray is the other category. The things that are included most often, so this is ranked from most often included to least often included, are adding an explicit incubating class for the mosquito, adding waning immunity, although that's not particularly common for wildlife infections, adding super infection or co-infection, that's primarily dominated by malaria and dengue, of course. And then following on from that, adding spatial structure, aquatic populations, non-constant per capita biting rates, multiple pathogen types, heterogeneous biting, non-constant per capita mosquito mortality, and non-constant transmission probability to mosquitoes, multiple vertebrate types. So that's particularly interesting for these wildlife diseases. There are certainly a number, it looks like about 10 out of the 26 West Nile models actually included more than one host species, which given that there are dozens of different competent hosts out there for West Nile transmission is a little bit shocking. Et cetera, on to the ones that are very rarely used. I also want to focus on blood meals on non-host species. There are very few models that incorporate that. Obviously, it doesn't make sense to think about blood meals on maybe all different non-host species, but certainly when you're thinking about what the effective biting rate is, the proportion that are biting on a host that's never going to infect them should be able to be discounted. That's not been taken into account in many models at all. So you could say that the conclusions of the Ryder, Perkins et al. Paper are blah blah, and future work, add heterogeneity, add spatial structure, add age structure, as Julia said, was basically the state of the art 20 years ago, at least for directly transmitted infections. This is the way they word it. Modern theory would benefit from an expansion around the concepts of heterogeneous mosquito biting, poorly mixed mosquito host encounters, spatial heterogeneity, and temporal variation in the transmission process. Meanwhile, while the modelers have been chugging along, reparameterizing the Ross-McDonald model and simple variations on that theme, empiricists have been busy measuring many relevant quantities, usually not in the context of a model or really often not even thinking about anything certainly more complex or more dynamical than just vectorial capacity, which does make it into the discussions a fair amount. But these data are often collected in a way that's completely unconnected from any sort of quantitative framework for interpreting them. And I'm just going to focus on three types of data that there's been a lot of focus on in the last 10 to 20 years, which isn't to say that people hadn't been also addressing these issues before. So mosquito biting behavior, which I'll talk about both experimental and observational approaches to host confidence studies, so host viremia and parasitemia profiles, and mosquito infection probabilities, which are generally thought of as vector competence studies. So we have experimental mosquito biting behavior studies and observational studies. So the distinction well, not the distinction I'm making, the distinction here tends to be that experimental studies are really trying to understand mechanism. To what extent mosquitoes exhibit preferences for taking blood meals from one species or another, or one type of individual versus another, and how those preferences are determined. Are they genetic? Are they based on their previous experience, et cetera? And observational studies are more phenomenological. So focusing first on the experimental studies. So you might sort of be skeptical of a lot of these studies if you're familiar with the literature. A lot of them have been done in a way that doesn't take into account the fact that there are multiple possible explanations for exhibited preferences. So they're measuring preferences, but they're not giving any information about the true mechanism underlying preferences. So this paper by Vladimir Alonso from 2006 reviewed the literature up until that time and talked about the ghosts that pester studies on learning in mosquitoes and guidelines to chase them off. And they highlighted a study that I'm particularly fond of and referred to it as a ghost busting study, which I particularly liked the term there. So I'm going to talk a little bit about this paper, which looks at host fidelity. So this is basically the idea that a mosquito will preferentially feed on the host type that it's taken a successful blood meal from previously. So this study, the way they set it up is in two stages. The first stage, they trapped mosquitoes that were either attracted to a cow in an experimental setup or attracted to a pig in an experimental setup. And then they placed these mosquitoes in an enclosure with either a cow, initially attracted to a cow is always in blue, initially attracted to a pig is always in red. And then they placed these mosquitoes in enclosures with cows, pigs, or both. And they then looked at what happened when they were placed into those new pens. So these are results for experiments where mosquitoes were placed in an enclosure with only one host, so either a pig or a cow. And the results show that individuals initially attracted to a cow are more likely to take a blood meal from a cow than a pig, and individuals initially attracted to a pig are more likely to take a blood meal from a pig. So that's fairly straightforward, but that doesn't get at that issue of the ghosts. So we don't have any mechanism to back up what could be driving this. It's just saying that the cow mosquitoes are cow mosquitoes, essentially, and the pig mosquitoes are pig mosquitoes. So here are some results for experiments where the mosquitoes were placed in an enclosure with one of each kind of host. So now they're given a choice. This is just additional evidence that the cow mosquitoes are cow mosquitoes. So data show that mosquitoes are initially attracted to the cow are more likely to choose to take a blood meal from a cow than a pig if presented with an equal opportunity to bite both, and that mosquitoes initially attracted to a pig are more likely to choose to take a blood meal from a pig than a cow. And most studies sort of stop there or don't even do that second, the choice experiment. But in this particular study, they decided to follow up to try to figure out whether this was actually based on prior experience or whether this was a genetic preference. So the follow-up experiment was actually had two goals. One was to examine whether this preference was observed if the mosquitoes were allowed to feed before they were trapped. So in that initial study, they were just in the vicinity of the host when they were initially trapped. They weren't given time to feed. And then the second goal of this study was to rule out the preferences determined by genetics. So in this study, mosquitoes observed to take a blood meal from a cow in a cowshed or from a pig in a pigsty were trapped, and their offspring were reared in the laboratory. The trapped mosquitoes were then placed in an enclosure with both a cow and a pig, and researchers measured how many mosquitoes took a blood meal on each host. Fairly straightforward. So the trapped mosquitoes' offspring were then also placed in an enclosure with a cow and a pig, and given the choice to choose on either to take a blood meal on either host. So this is the results for the parent generation. So the trapped mosquitoes that had previously taken a blood meal from one of the hosts had a clear preference for the host species where they had taken that previous blood meal. That's what you would expect based on the first set of experiments. And of the mosquitoes that fed in the experimental enclosure, all mosquitoes trapped after feeding on a cow chose to bite a cow. Some of them also bit pigs. And of the mosquitoes that fed in the pigsty, most of them chose to bite a pig. Some of them chose the cow instead. So then what happens with the offspring? So the offspring are shown here. And you can see that the offspring of trapped mosquitoes showed a very different pattern, indicating a preference for feeding on the same, indicating that the preference for feeding on the same host species as on the previous bite was not due to a genetic predisposition for biting that species, and was likely based on previous exposure. So the offspring of mosquitoes trapped after taking a blood meal were more likely to bite cows than pigs, independent of whether the mother had taken a blood meal on a cow or a pig. In general, this species, so I should mention that they actually did this with 4 different species, and I've focused on Culex vishnui just because I felt like presenting all 4 of them would be a bit exhausting. Essentially, the mosquitoes appear to prefer to take blood meals from the same species as their last blood meal, which is a behavior we would call fidelity. That's the experimental side of certainly, one study to characterize many that have occurred in the past 20 years. And the observational side, as I mentioned, is more phenomenological and essentially consists of field studies of blood meal distributions among hosts that measure the outcomes of this process without trying to understand the mechanism underlying it. And there's been a lot of recognition, particularly in the last 10 years, and focus on West Nile virus has definitely brought this up. And certainly, similarly, I would say for tick-borne diseases as well, including Lyme disease. But there's been increasing recognition of spatial and temporal variation in biting patterns. And just one example is, so these are Culex torsalis bites through time at a heronry in Davis, California. So you, as one might expect, being in a heronry, they tend to bite primarily herons and egrets, which also are common in this heronry, through much of the year. So basically April, May, June, July, August, September. And then most likely driven primarily by the abundance of different bird species, switch to very different hosts, including the American robin, but also including things like the western fence lizard in small numbers, and a variety of mammals up here in green. So very clear temporal patterns. How this relates to density of the hosts is a matter of some debate, whether it's purely driven just by availability or whether it's driven by some of those types of preferences that I was talking about before. So there's been a lot of measurement going on. I would say that little's been done by way of incorporating these patterns into models and examining their impact on dynamics. Often people do try to parameterize either R naught or vectorial capacity and make a statement about, without taking in, most often, there are some exceptions, but in the mosquito literature, most often, there's very little discussion of uncertainty or variation, and all the numbers are sort of pulled together from various different places, and then a value of R naught is proclaimed, which I'll get back to in a minute. Okay, so moving on to mosquito infection probabilities. So there's been a lot of experimental studies. This is just 6 with fairly good data. But typically mosquito infection studies are measuring either the probability of infection or the probability of a disseminated infection or the probability of transmission, various different ways to assay those different endpoints. As a function of something like the infection dose, the temperature of when the dose was given, the influence of larval habitat has been something that's been starting to take off, people have an interest in. Certainly the time since exposure and strain differences. Very, very rarely do people actually look at multiple variables, multiple variables at the same time. And I think that that's something that is is needed in the future. So these here are dose response data, and dose response data, usually the way they're used in the medical entomology literature or the veterinary epidemiology literature is that they make these very nice curves which you can fit with mechanistic or pseudo-mechanistic models. So these are fit with essentially a dose-response model like is often used in gastroenteritis dose-response modeling. But typically in the medical entomology literature, they're used to determine a threshold value, and it's then sort of arbitrarily cut off at that value, and anything below that value is considered not to infect mosquitoes, and anything above that value is considered to infect mosquitoes. And that is somewhat convenient in that it allows really easy calculation of various indices of host competence, where you can sort of rank species in terms of how important they might be for transmission. But it's brushing a lot of the details under the rug, obviously. And then lastly, host viremia and parasitemia profiles. And this is some viremia profiles for 9 North American bird species inoculated with 2 different strains of Japanese encephalitis virus. And this is the sort of study that people do when they're sort of concerned about a pathogen coming into a new area. They say, well, what hosts do we have? What potential hosts do we have? What potential vectors do we have? And is it likely that any of these are competent host-vector combinations? And as I said, these are typically used in combination with those sort of dose-response curves to say, well, if, say, our threshold is 3, then there are 2 days with this strain where the house finch is above 3, and therefore it has an infectious period of 2 days. And following on from that, calculation of competence indices. I would argue that other than some of the mosquito biting behavior, none of this is really new. People have been trying to do these things for a long time, but there's been a lot of new sort of molecular developments that make the assays better and add precision to the measurements. And there's been a large buildup over the last 30 years or so of these types of data that I think are sort of ripe for systematic approaches to be used on them. And I've mentioned several times that occasionally these data are collated somewhat systematically and quantitatively either to estimate vectorial capacity or competence index or something along those lines, or not. So that was a multi-host version of R naught. And this sort of brings us back to things that Christai was thinking about in the late 80s and early 90s. And at the time, he argued that not all of these quantities can be measured precisely enough to give robust calculations of R naught. So we may spend a lot of time and effort measuring these different quantities, but we still are really unlikely to ever know whether R naught is greater than 1 or less than 1, even at that level. So at the time, he was arguing for thinking more about relative measures. And so this general conclusion, basically being the conclusion that we're never going to be able to pin everything down precisely, suggests a shift in emphasis in quantitative studies of transmission of insect vectors. The investigator should tackle more explicitly comparative questions, such as what is the most important vector, what is the season of transmission, when is the maximum, what explains the geographic variation and prevalence of infection, et cetera. And I would argue that a lot of these things are maybe better understood now than they were 20 years ago, certainly for malaria and dengue, but probably not so much for especially what explains geographic variation in the prevalence of infection, something that's a big, wide open question for most of these multi-host pathogens, multi-host vector-borne pathogens. And I would also argue that there has been a large buildup of data in the literature, and I think it might be time to start thinking about the interactions. And some work, again, has been done on this so nice work by Andrew Reed and Matt Thomas and folks like that on malaria, but very little work on multi-host pathogens. And particularly, I think it's sort of time to start rethinking about what we mean by competence. So competence has been sort of a measure that is supposed to be the intrinsic ability of either a host or a vector to become infected and produce an infection. And I think that these things are really, there's evidence coming online that these things are really context dependent. And this is just a simple example where we have two species, one with a high peak viremia, one with a low peak viremia. And at two different temperatures, we have slightly different curves for the probability of infection, not even taking into account the follow-on effects of the probability of infectiousness, et cetera, as a function of the dose received. And even just with a difference of 1 degree C, you can obviously see that as a function of mosquito to host ratio, the proportion affected doesn't change very much for the high viremia. Sorry, the colors are a little confusing, but the high viremia host is essentially maxed out by 27 degrees C, and with even a very low mosquito to host ratio can become endemic. And the low viremia host here would have a very hard time establishing at the lower temperature, but just a single 1 degree increase would essentially move the threshold substantially. And the reason that I'm thinking about this, and the reason I think it's important, is I think that particularly for Japanese encephalitis, which is one of my main areas of interest, there are regions where the sort of transmission dogma of pigs being the important amplifying host, which is driving all of the human infections, it really just doesn't match up with the geography of where the disease has recently been found. And so there are lots of areas without pigs at all, and we know that there are plenty of other hosts out there, birds that can be infected, but they tend to have much lower viremias. And I've started thinking a lot about sort of how the the environmental component comes into play in terms of tropical regions like Bangladesh, obviously are very different from the places where it was originally studied, such as Japan, where these differences in temperature are very realistic, and really you could see a completely different suite of hosts driving transmission and at least driving maintenance in these regions compared to the places that these things have traditionally been studied. And with that, I'm going to turn it over to Andy.
I think in the interest of time, we can just switch over straight to Andy and then